guys, today we will be discussing about the cholinergic transmission as we discuss the physiology of the sympathetic and parasympathetic system. Now it's time to deal with cholinergic transmissions, and further we'll continue with adrenergic and then dopaminergics and further on. Now the cholinergic receptors and the signal transmission is done in a very specific way. But let's start the lecture in this way that first we will synthesize the acetylcholine. So we'll discuss about the synthesis. And then let's move and see how the acetylcholines are stored. Then we'll discuss how the acetylcholines are released. And then we'll continue with how they are reuptaken or metabolized there, so the reuptake there. And after that we'll discuss about the types of acetylcholine receptors, and after that we'll discuss what is the tissue response, and the pharmacology we'll deal in another lecture. So let's get started with synthesis, and then we'll continue with other sections. So in order to explain you the synthesis, let me just clean the board here and move this diagram into the center a bit, so you can see it much more better. Okay, this is a presynaptic junction or presynaptic cell. Now in presynapses there are very special transporters. Example, there is a very special transporter for choline, not acetylcholine but choline molecule, which is taken by diet. Imagine this is for choline. And then there is a very special another transporter for calciums, not potassium but a calcium. Especially this transporter is dependent on voltage, so this is a voltage dependent calcium channel. So let's see from where the choline comes and enters and how they are going to form the acetylcholine. Now the choline, as we said, it is taken by diet, and as it enters the presynapses here, of course it should be converted into acetylcholine. In order to convert choline into acetylcholine, we need of course acetyl molecule. So the acetyl molecule could be seen in mitochondria. So here's our mitochondria, which is all the time releasing acetyl-CoA. So from the mitochondria there will be release of acetyl-CoA in this term. And from the channel of choline, this is the choline transporter here. Please understand and memorize transporters and all these steps carefully, because there's a lot of molecules which can inhibit the transporters and reactions. So the choline is now in the cell inside of the axonal ending. So this is our choline molecule. Now what's going to happen is that there's a very special enzyme which is called acetylcholine transferases, and the acetylcholine transferase is going to take the acetylcholine happily and going to bind it with acetyl-CoA with acetylcholine and releasing the CoA group from here. So as they bind with each other, as they join with each other, there will be formation of acetylcholine. This is how the acetylcholine is synthesized by the help of acetylcholine transferases. And in this reaction, the CoA group was removed and released. Now let's move on. Let's see how the acetylcholines could be stored. Now for storage of acetylcholine, we need some special types of vesicles. So this is a very special vesicle for storage of the acetylcholine. Now on the surface of these type of vesicles, there are very special transporters. These transporters are called acetylcholine vesicular transporter. So this transporter is going to take the acetylcholine into the cell. Let me name it vesicular acetylcholine transporter, VA acetylcholine transporter. Now what's it going to do? It's going to bind, it has a very high affinity to acetylcholine. It's going to bind with the acetylcholine and it's going to take the acetylcholine into the vesicle. So here's our ACH in the vesicle, happily waiting for release. Now the release is, of course, it takes also some steps, but before that, let me explain you that there's also some other transporters such as ATP transporters, vesicular ATP transporters here. So some amount of ATP will also be stored in this section through the ATP transporters, or vesicular ATP channels transporters. So this is how the ATP and vesicular acetylcholines are formed and stored. Let's see how the acetylcholine could be released from here. Before going to that, let me tell you some specific things about this vesicle, because on the outer membrane of this vesicle there are very special proteins located. Now these proteins, which are seen in the outer surface of the vesicles, they are called snare proteins. Now the snare protein is actually an ethanolamide sensitive factor. So vesicular ethanolamide factor. Now there could be two different types of snare protein, which could be vesicular snare protein and the membranal or neuronal snare protein. Now the neuronal snare protein are located, as you understood from the name, on the membrane of the neurons here. So let me just show it to you. Here it is. These are snare proteins for neurons. Now, of course, they're different from vesicular snare proteins. Example, snare proteins. We have synaptogmeins. Synaptogmeins, and also there are some such as synaptobrevins. And there are neuronal snares such as SNAP25, or there is one more which is called syntoxin 1. Now, to know them also important because the vesicles will come and will bind with these snare proteins via snare proteins of the vesicles, so they could be released from here. And let's see how they can bind with each other in order to release the acetylcholine. Actually, there's a very little problem here. Let me just extra zoom here so you can understand much more better. Now here's our snare vesicle, which is full of acetylcholine and ATP. And as I said, on the snare proteins, there is on the vesicle there is a snare protein. So also let me mention that the snare proteins actually they have negative charge, so they are negatively charged. Also, there are snare proteins on the membrane here, and we just named them neuronal snare proteins. The neuronal snare proteins are also negatively charged. So the negative charge, with negative charge, they cannot bind with each other. Oppositely, they push each other. So we need some other molecule in order to join them, so the vesicle can diffuse on the membrane and release its product. Now, in order to do that activity, we need action membrane potential. As the action membrane potential is generated, the action membrane potential, of course, goes through the membrane. So it's coming like this, and just continues on the neuronal membrane. But whenever the action membrane potential reaches to the voltage dependent calcium channel, actually it's going to activate this channel, and from here there will be a leakage of thousands of calcium molecules. So as the calcium molecules now enters the axonal ending, they can make some change. Now let's see which kind of change they can make. Actually, those calcium which entered by the help of action membrane potential to the neuronal endings now, they can bind with snare proteins in the vesicles, and they can bind with snare proteins in the membrane, also from other side. Now, as they bind with each other, these calciums are helping snare proteins to get close and attach with each other. Okay, as they attach with each other, now let's see, this is going to happen, this twisting is going to happen. As this coiling is continuing, actually the vesicle gets closer to the membrane. Yeah, that's what was my charger. So let's see what happens here. Now, here is our vesicle, let's say, and this was the snare protein of the vesicle, and this was the snare protein of the neuronal membrane. Again, the same thing here, and the membranal snare protein. So they joined with each other, and they started moving like this, coiling and twisting like that. And as a result, the vesicle got closer to the membrane, and as we know that both of them has phospholipid membranes. So the lipid membrane, they fuse with each other, and in that fashion, this area got open. It gave chance to vesicle to release its products. So here we had previously these molecules of acetylcholine and ATP. So those are now released into synaptic cleft or synaptic junction. Now from here, what's going to happen? Let's see. Okay, as you see these blue dots, they are acetylcholine molecules. Actually, on the postsynaptic junction, there are very special enzymes located, usually in the synaptic cleft, and these enzymes, they are breakers. They are going to break down the acetylcholine which came here. Now the acetylcholine is here on the synaptic cleft. It has to go and activate, of course, the acetylcholine receptors, which we'll discuss in some minute. So here are the receptors first, where the acetylcholine will bind with them, and then they will activate the intracellular signaling, so there will be tissue response. But before that, the acetylcholine here, which is in the cleft synaptic junction, is going to be catabolized, broken down by the help of this specific enzyme. Now this enzyme, it is called acetylcholine esterase. Acetylcholine esterase is going to break down the acetylcholine into two molecules, and that is the choline and acetate. So the choline again will recycle and will enter through its cholinergic channel like this here to presynapses. So there it will continue its circulation, and the acetoacetate will be further on broken down and added the acetyl-CoA and so on. So this is how the acetylcholine is released to the synaptic junction. And now let's see which kind of receptors we do have and how they are going to act and activate which kind of intracellular signaling pathways. So let's move on forward. So before moving to receptor type and tissue response, let's discuss about some blockers which can inhibit these pathways. So primarily, the choline transporter could be blocked here. There are some agents which can block the choline transporter. So here, I mean, if the choline transporter here it is blocked, of course the choline will not enter, the acetylcholine formation synthesis will decrease. And this kind of molecule here which blocks that, it is called hemicolinium. This is a acetylcholine transporter blocker. And then other blocker could be seen in between the snare proteins here. As you see, as the calcium comes and try to bind the snare proteins with each other, there is a toxin which inhibits this blockage, this attachment, and that toxin is called botulinum toxin. Botulinum toxin. So the botulinum toxin is actually blocking inhibiting the attachment of snare protein with snare protein in the neuronal and vesicular side. And also we can have other inhibitors, such as inhibitors for acetylcholine and esterases, and also here it is. We can also inhibit this entrance of acetylcholine to the vesicle, so we can block the vesicular acetylcholine transporter, and that transporter is blocked by a molecule which is called, yes, the molecule which is called, if you're thinking about Vesamicol, you're absolutely right. Vesamicol, let me write it more understandable. Vesamicol. So the Vesamicol is going to inhibit the acetylcholine transporter, which is located on the vesicular membrane. Now, what else? Which other kind of blockers we can discuss? We'll go to detail of acetylcholine esterase inhibitors later on. And what else I can tell you? Okay, now let me tell you that if we inhibit the choline transporter, the action is going to happen like this, that the acetylcholine synthesis will decrease. But if we block the vesicular acetylcholine transporter, in this case, again, the number of acetylcholine storage will decrease and the function of acetylcholine decreases. And if we block the snare proteins there between the vesicle and the membrane, the neuronal membrane, again, the release of acetylcholine will be problematic, so the acetylcholine again decreases. But there is one thing that you should know. If we block acetylcholine esterases, what's going to happen? That in the synaptic junction, the accumulation of acetylcholine will increase and the activity of acetylcholine increases. So this is the only molecule here in this section which increases the acetylcholine activity by increasing its numbers in the synaptic junction. So this is some blockers and normal physiology of acetylcholine, how we synthesized, how we stored and released. Now we will move to types of receptor and their action. So let's move on. The acetylcholine receptors, primarily it could be divided into two main categories. And those are nicotinic receptors, acetylcholine nicotinic receptors, and muscarinic receptors. So this is the two main type here. Now, how they differ from each other? Actually, the mechanism of action of them is slightly different. The nicotinic receptors, when the acetylcholine comes and binds, they act through other mechanism, and muscarinic through other mechanism. Now, let's briefly see how they are acting. Example, this is acetylcholine here. It came and bind with nicotinic receptor. As it binds with nicotinic receptor is going to open channels. So it's going to stimulate the specific channels so they can open. Opening the channels. And all I can say that it is the nicotinic receptors are usually ligand-gated ion channels which they are activating. So here we have these ligand-gated ion channels. And what about the mechanism of action of muscarinic? That's slightly different, as I said. Example, here it is the acetylcholine, and this is the muscarinic receptor. As the acetylcholine binds to muscarinic receptor, it's not going to open any channel itself, but it's going to initiate a cascade of reaction in the cellular membrane. And that cascade of reaction will bring to thousands of change in the cell. And that cascade, or the type of intercellular signaling which muscarinic receptors are bind, it is G-coupled proteins. And the G-coupled proteins we discussed in intracellular signaling. So if you didn't watch them, go and watch. It's very important to watch them before everything, I think. So this is how muscarinic receptors are going to work. After they stimulate the G protein, there will be initiation of cascade, and at the end there will be activation of protein kinase C and calcium kinases, and so on. Now let's go to detail of these receptors and see really how they act actually. And let's get started with nicotinic receptors, and then we'll move to muscarinics. Now, the nicotinic receptors, further on, they could be divided into two small subunits, and they are nicotinic receptors which are located in the muscular cells, so nicotinic muscular subtype, and nicotinic receptors which are located on neurons, so nicotinic neuronal type. And they both activate the channels. The mechanism of action is very same, but the structure of these subtypes are slightly different. So let's go and see each of them. Now let's discuss with nicotinic muscular. Now imagine this is a muscular cell membrane, where the okay, let me make it by phospholipid membrane. So here is the postsynaptic junction and release of acetylcholine, and there is a channel which we want to discuss about, and that channel is for sodium and potassium calciums. Here is our sodium potassium calcium channel. Now so primarily it is not open. Now the main function here is actually to open this channel, so there will be generation of action membrane potential in order to continue the activity. Now what happens, that the muscular type of nicotinic receptor, further on it could be divided into two main small category. So let me divide that. So here we have this nicotinic muscular unit. Actually, the nicotinic muscular unit is a little bit different in adults and in infants. So it's slightly different in infants and adults. Let's see what are the main differences here. Actually, the main differences in structure of them. Now let me show you one thing, that the nicotinic muscular subtype receptor is made up from 5 subunits, and that is alpha, beta, delta, gamma, and epsilon. Now, it's the main structure of muscular type of nicotinic receptor. But let's see what will be the difference between adult and infant one. Now, let me try to draw you these receptors. Now, here is our infant nicotinic receptor, and let's say in this side we have the adult nicotinic receptor, which is the muscular type. Now, the adult nicotinic receptor is made up from alpha subunit, beta subunit, and a delta subunit. And the main subunit here, which differs this from infant, it is the gamma subunit. Now, epsilon subunit, actually the gamma subunit is in infants. So this is the epsilon subunit. Alpha, beta, delta, and the epsilon. And in infant acetylcholine nicotinic muscular type receptor, it has alpha subunit, it has a beta subunit, as well it has a delta subunit. But the other part, or the last subunit here, is now the gamma subunit. So this is the main structural difference between the adult nicotinic muscular type of receptor and infant nicotinic muscular type of receptor. It doesn't matter which type, but the result will be the same. Example, the acetylcholine, the acetylcholine here comes, and let's say it comes to the side as well, and let me put here some dot. Now as the acetylcholine comes and binds to the receptor here, now it's not binding to the G point, the gamma point, but it's binding overall the receptor, and here also it comes and binds. As they bind to this nicotinic muscular type receptor, what happens is that they stimulate the channel, and the channel gets open. As the channel gets open, from the channel there will be leakage of hell of sodium. So sodium, potassium, and calcium can enter through this channel. As they enter through this channel, of course, they will change the intercellular charge here, these molecules, and as a result there will be provoking the action membrane potential. And the action membrane potential, we will discuss what it will do here. So let's move on to other type. So let's move to nicotinic neuronal type now. Here it is, as we discussed the muscular type. Now the nicotinic neuronal type is having the very similar mechanism of action. So I'm not going to detail of that because the mechanism of action will be same. The acetylcholine will come and binds the receptor. The receptor opens the channel. From the channel leaks the sodium, potassium, and calciums, and it provokes the action membrane potential. This is the same mechanism of action. But the only difference between muscular and the neuronal type is, of course, the location, plus location, the structure of them. So let's discuss a little bit about the structure of them. Actually, the nicotinic neuronal type is further on is made up from two different small subunits, and those are alpha subunits and beta subunits. As you remember, the muscular type were made up from 4 subtypes here: alpha, beta, delta, and epsilon and gamma. But here, the neuronal type is only made up from alpha and beta. But you should know that there is more than 9 type is identified in alpha, so there is 9 different type of alpha subunit and 3 different type of beta subunits, which are beta 2, beta 3, and beta 4. And the alpha is alpha 1 up to 9. Now these are some different types of structure in the neuronal subtype. Now here you should know that these receptors are locations of these neuronal subtype, because the neuronal subtypes could be located in different sections. Example, let's say the neuronal subtype which is located in peripheral nervous system. Peripheral nervous system. There we have nicotinic neuronal subtype of beta. Wait a minute. We have a alpha 3 and beta 4 subunits, and in central nervous system, the receptors of the nicotine neuronal type is usually the alpha 4 and beta 4. Here is also beta 4. So this is only difference and in locations as well. And about the poisons and other drugs which can inhibit or stimulate these drugs, these receptors, we'll talk in a minute. So this is all about the nicotinic acetylcholine receptor subtypes. Now let's move to muscarinic subtypes, which is more than nicotinic. So here is the muscarinic subtype now. Now the muscarinic subtypes are further divided into 5 small subunits. Yes, they are M1, M2, 3. The most important is M1, M2, and M3. because they can be seen in several different sections of the organism. But M4 and M5 is usually located in central nervous system, but the remaining could be seen in peripheral system as well. Now let's discuss a little bit about their differences with each other. Now, example, the M1 is usually located, of course, in the CNS. It could be found in CNS. Plus CNS, also it could be seen in a lot of glands. What concerns M2, it could be seen mostly again in the CNS, as well as in special smooth muscles. So we'll be seeing what kind of effects will bring the M2 if we stimulate them in smooth muscles. And the M3 is pretty much seen everywhere else, I can say. It could be seen in lung, it could be seen in heart, it could be seen in all other tissue. So it could be seen in all other tissue. But M1 and M2 is more specific for smooth muscle and central nervous system. What concerns the M5 and M4 here, they're both predominantly seen in central nervous system and in specific areas of central nervous system, such as substantia nigra and others. So let's discuss mostly the function of these, what will be the effect of these stimulation of the M receptors. Now I'll give some specific examples here. Example, the M1 is intracellularly, it's attached with GQ protein. And we know that the GQ protein is a stimulatory protein. So if we stimulate the M1, it's going to activate something. So it's a stimulatory pathway. As a result, if we take an example here, let's say that they are located in glands, and we stimulate M1 in glands, and the result was activation of a GQ pathway, and the result of the activation of GQ pathway would be increased in secretion. So this is how it affects the gland. And what concerns M2, we say that it could be seen in CNS and smooth muscles. Let's take example of heart. And in the heart, the M2 receptors could be seen in SA node or AV node. But the question here is the stimulation of M2 receptor by acetylcholine. The intracellular signaling here is going to be slightly different than M1 because it's going to activate the GI's inhibitory pathway. So this is an inhibitory pathway which is activated by stimulation of M2. And if you know, as you know, if we stimulate the M2 in the SA node or AV node, it's going to decrease the heart rate and conduction in the heart. So by stimulating M1, we can decrease the heart rate here. So let's move on to N3 and give some examples here so you can understand much more better. Now the N3 is it's working again through the GQ pathway, which is stimulatory pathway. Do not forget and do not confuse, guys, that in muscarinic receptors, some of them are coupled by inhibitory pathways and some of them are coupled by stimulatory pathway. Let me actually mention them here so you will not confuse. That M1 is coupled by GQ, which is a stimulatory pathway. M2 is coupled by GI, which is an inhibitory pathway. M3 is coupled by GQ, which is stimulatory pathway. M4 is actually coupled by GI, which is inhibitory, and M5 is a GQ, which is a stimulatory pathway. So please do not confuse these because they are very important to understand their functions. And before, if you are listening to this lecture first time, and if you didn't listen or didn't study about the G-coupled protein intracellular signaling pathway, please go study that topic so you can understand here much more better. Now, let's discuss about some organs which are located, which the M3 could be seen there. Example, let's take eye. If we stimulate M3 receptors in the eye, we are going to stimulate smooth muscles there, and let's say we stimulate the sphincter, or we stimulate the ciliary muscle. Now, if we stimulate the sphincter muscles here, the eye will go under, these muscles will go under contraction, and due to contraction, there will be meiosis. And if we stimulate the ciliary muscles, again, they will go into contraction, because we say that it's a stimulatory pathway. And as contraction, it helps the people to accommodate for near vision. I mean, if something is getting closer, it means that the acetylcholine will be released, and they will stimulate the M3 receptors in ciliary muscles, and there will be contraction of the ciliary muscles, which helps us to accommodate for near vision. So let's say example, lung. In the lungs, we also have M3 receptors, especially in bronchioles and glands there. Bronchioles. Now, if we stimulate M3 in the bronchioles, again, it will bring for contraction, because it's a positive stimulatory pathway, and the contraction in bronchioles will bring to what? Bronchospasm, which is not a good idea to have in asthmatic patients. Or let me give you some other examples, such as if it is located in bladder, urinary bladder, I'm talking here. Now, if we, if the acetylcholine goes and binds with muscarinic receptor there, of course that is M3, of course that is G coupled, and that is Q coupled actually, and that's a stimulatory. And there will be contraction of and muscles, and those muscles such as the trousseau muscle, the trousseau muscle, and also it can act on other muscles such as sphincter muscles. And if the acetylcholine comes and acts on them, in the trousseau muscle it brings for contraction, and in other muscles it can bring for relaxation or dilation, and due to that reason it can cause voiding, or it can cause the urinary incontinence. It's like this, this is a urinary bladder, let's say. Okay, it's a bad diagram here. Okay, I'll just draw it like this. It's a urinary bladder. Here the urine must go, and here is the sphincter and the trousseau muscle located around the urinary bladder. As you see here, if we stimulate the trousseau muscle, it's going to contract. It's going to contract the bladder, and if the acetylcholine stimulates the sphincter here, it's going to relax. So this area gets open, and from upper side the bladder gets contracted, and there will be release of urine incontinently. This is some example how they are working. We'll more discuss about the types of receptors in pharmacology. We'll see which kind of pharmacological agents act there, block there, and stimulate them. So this is all about the cholinergic transmission, their receptor types, and storage synthesis, and some information about the tissue response. So next time we'll discuss about the pharmacology. Until then, goodbye.